Dear students, this is the 8th video of the Palynology series of lectures. In this video, we will talk about the chemistry of sporopollenin. In the last videos, we discussed a little bit about sporopollenin, that it is a very tough biopolymer in nature. It is probably the toughest one. No artificial or natural chemical substance, organic molecule is as tougher as sporopollenin. Because of its chemistry, it is very complex molecule. In this particular lecture, I will discuss a paper which was published in 2018. So it is probably the latest paper about sporopollenin chemistry, published in Nature Plants by Fu Shuang Li et al. This plant discusses the chemistry of sporopollenin of Pinus rigida pollen grains because of the availability, because this particular pollen grain is produced in enormous amount, and so you get enough samples to do your studies, to do your NMR or HPLC or whatever you want to do, or circular dichroism. Different type of techniques are applied to understand the structure, composition and structure of the chemical substances, the monomers that actually make the sporopollenin biopolymer. Sporopollenin is so tougher that it is resistant to any type of acidic or alkaline environment. It is also thermostable, probably because this exinous wall that protects the gametes that are present inside, that are carried to the stigma and then followed by pollen tube germination. So sometimes when pollinators carry the pollen grains, they have to travel long distance. So pollen grains have to face different kinds of environmental assaults. So to this exinous layer comprised of sporopollenin protects the pollen grain from environmental assaults. Land plants evolved 450 million years ago, and then a switch from the aquatic habitat to land habitat occurred, and at that time there were so many different kinds of co-evolution that took place that we know that the vascular bundles that developed, the root and the stomata and the cuticle, all these things, the morphological changes that occurred for adaptation to the terrestrial environment, which is comparatively harsher. There were so many biotic and abiotic stresses that are present in the terrestrial environment, so plants have to face this. So because of that, some chemical changes, some chemical coevolution also took place. Molecules like lignin, condensed tannin, you may talk about cutin, suberin, natural rubber, all these molecules, and along with this sporopollenin, these molecules got evolved. And what was the biochemistry behind this? What biochemical pathway actually led to the synthesis of this kind of molecules is very difficult to assess, particularly with respect to sporopollenin. Polyketide biosynthesis probably is the mechanism by which sporopollenins are synthesized, but still the mechanism is very ill represented, and it is very elusive. So it needs to be studied further, that what is the bio, but it is definitely the derivative of polyketide biosynthesis. So in this paper, they have done in thioacidolysis of sporopollenin and then followed by HPLC UV mass spectroscopy and then NMR studies to understand the different components of sporopollenin. So far, this is probably the best analysis, as I can say. Way back in 1937, when Jesche, he gave the empirical formula of sporopollenin to be C90 144O27. After that, there were so many different studies, but none of these studies could decipher to this extent. Here, after doing the thioacidolysis, Now, before, let me describe the process a little bit. After getting the pollen grain, what they are doing at their, they say they are doing a ball milling, which is basically grinding into powder, and then followed by some enzymatic degradation, digestion, and wash. They go for thioacidolysis. This results in two components: one soluble component, another insoluble component. The soluble component is subjected to HPLC UV mass spectroscopic analysis, and this analysis results into different kinds of chemical substances in the pollen grain. The most predominant among them is the one type of polyketide derivative, that is polyvinyl alcohol, which is quite a long chain molecule being synthesized from polymerization of the smaller molecules. The next molecule, predominant molecule that was found, was the para-Coumaroyl 7-hydroxy aliphatic chain with two, at the termini there were two functional groups. Those are dithioethyl groups. So this is the core molecule which is present in the sporopollenin, which connects the C38 long chain PVA, polyvinyl alcohol, which are also linked together by ester or the ether bonds. So this is the molecule in which both ester and ether bonds are present. Other chemical substances like lignin, tannine, suberin, cutin, etc., they have only one, either ester or ether bonds. That's why this is probably the most difficult molecule to degrade. Okay, so what about the other molecules? There are, so here if you see the chromatogram, the most predominant one is this para-Coumaroyl 7-hydroxy aliphatic chain with the dithioethyl groups. The next molecule which was predominant was 2A plus B. This A plus B, they came together in the chromatogram. They could not be separated because of the similar nature. They are derived from 1. Basically, they are the hydroxyl substitutes of 1. In one case, the carbon 1 position has a hydroxyl group. In the other, carbon 16 has a hydroxyl group. It has replaced or substituted the dithioethyl functional group of the para-Coumaroyl molecule. So if you talk about the predominancy of these two molecules, 1 versus 2A plus B, the ratio is about 6 to 7 is to 1. So about 6 to 7 times this dithioethyl derivative of long fatty acid chain is present in comparison to their hydroxylated derivatives. Now there were some minor components like the para-Coumaric acid, some dioxane molecules, and also some naringenin molecules were found. If you see this diagram here, this is the model structure of sporopollenin after doing this kind of studies, HPLC, NMR, and other things, circular dichroism, and many other things. I'm not going to discuss all those difficult matters here. They led to the designing of this particular model structure of sporopollenin, which says that there are long chain molecules of C38 long aliphatic chain, which comprises at the one end, it is basically, this is the molecule which is polyketide derivative polyvinyl alcohol, which at one end contains alpha pyrrone, at the other end it contains an ester linkage which is connected with the next molecule. So like this, these are running parallelly and they are interconnected by the para-Coumaroyl C16 aliphatic chain by ester or by ether linkages. Some para-Coumaric acid molecules and some dioxane molecules are linked with this para-Coumaroyl interconnector. Those are also present. Naringenin is not shown here, but it is also present quite a bit amount in the sporopollenin. So this C38 molecule is basically produced from a C8 structure by cyclic synthesis, which has not been actually deciphered how it is being synthesized, and this may vary. The number of C38 may vary from species to species. And the complete structure, this structure has been given with respect to the pollen grain of Pinus rigida. There are others where also you can find there may be differences, of course. So this cannot be said to be the complete structure. However, through this video lecture, I tried to make a comprehensive analysis with the help of this paper, that how far we have known about the chemistry of sporopollenin. When further update will come, I will tell you further research after maybe. It is very difficult, that this molecule is so difficult. And I will request you to press the like button and subscribe and share to your friends. Thanks for watching.